Welcome back to Crash Biology in Stephen classes. In the last module, we have already discussed 10 chapters. From today onwards, we are moving with the second module having 10 chapters again. We will start discussion of a first year chapter, circulation in animals. Ready? So here we go. Circulatory system. You know, in the case of animals having a circulatory system, their circulatory system is of two types. Sometimes it will be open and sometimes it will be closed. What is the difference between open circulatory system and closed circulatory system? In the case of open circulatory system, their body fluid will be flowing through some open channels called sinuses. So all the visceral organs will be bathed in blood in them. In the case of closed circulation, you can see the body fluid flowing through blood vessels. You can see the regulation of the blood flow because of the valves that are present in closed circulatory system. So here the blood flow is very much regulated in closed circulatory system. And the efficiency of blood flow will be higher in closed circulation compared with that of open circulation. Right. Now what about the animals having these two different types of circulation? When you consider the animal groups, open circulatory system is applicable for all arthropods. All arthropods are having open type of circulation. Non-cephalopod mollusk, you know, phylum mollusca is classified into gastropoda, pelecypoda and cephalopoda. So here cephalopoda group is having closed circulatory system, but non-cephalopods are having open circulatory system. Hemichordates have open circulatory system. So these are the animals in general having open circulatory system. What about closed? Closed circulation is applicable for vertebrates, cephalopod mollusks, annelids and echinoderms. So the animal groups you have to remember always, and also the different examples coming under these animal groups you should by heart to answer the questions coming from the two types of circulation. Ready? So here we have a question for you. Identify the animals with open circulatory system. Open circulatory system is applicable for different types of animal groups. All arthropods are having open circulation. Like that, non-cephalopod mollusks are having open circulation. And also hemichordates are with open circulation. So we have a set of examples now. Azidia, cockroach, earthworm. You know, Azidia is having open circulation because it is coming under Urochordata group. Then cockroach is also having open circulatory system. Earthworm, it is an annelid. It will have closed circulatory system. Prawn is a crustacean coming under Arthropoda. So it will have open circulatory system. Silverfish is again an arthropod. Lepisma, that is a scientific term for silverfish. It will have open circulatory system. Snail is having open circulatory systems. Squid is having closed circulatory system because it is a cephalopod. Right? So here earthworms and squids, they have closed circulatory system. So our Acidia, cockroach, so 1, 2, 4, 5, 6. 1, 2, 4, 5, 6. I mean option D will be our answer. Right? So correct answer will be option D here. You have the answer shown here. So shall we move to our next question? So here we have the next question on the screen. It is about the components of blood. We have different components of blood. You know, blood is having a fluid part which is called plasma and also formed elements. Formed elements, by that we mean the blood cells. So here, what are the different formed elements you can see in our blood? There will be erythrocytes, also called RBCs. Leukocytes, which are also known as WBCs. And thrombocytes, also called platelets. RBCs, their shape and their function. You know, RBCs, they are concerned with, they are biconcave in shape. They are concerned with the transport of gases. Their mature RBC will not have a nucleus in it. What about WBCs? WBCs can be classified into two categories, granulocytes and agranulocytes. Granulocytes, they have granules in their cytoplasm. You can see it very clearly that granulated cytoplasm is applicable for granulocytes. Examples are neutrophil, eosinophil and basophil. So these are the examples for granulocytes. And you know their function. Neutrophils are phagocytic in their function. What about eosinophils? They can involve in allergic reactions. Basophils, their function will be secretion of serotonin, heparin, histamine, etc. So that is a function of basophil. What about agranulocytes? Agranulocytes are having a clear cytoplasm. They don't have granules in their cytoplasm. Examples are monocytes and lymphocytes. So both of them, they have a peculiar function. They are involved in immunity mainly. Actually, the lymphocytes are involved in immune responses. Phagocytic function is applicable for monocytes. So that is about our classification of blood cells and also their function. Platelets, you know, platelets, they are also called the thrombocytes. Their function will be in blood coagulation. They are involved in blood coagulation. Right. So we have the question now. The question is based on the function of these blood cells. Match the following and choose the correct option. So here we have the options for you. RBCs. RBCs, their function will be transport of respiratory gases. So what about neutrophils? Neutrophils are phagocytic in nature. Monocytes are also phagocytic, but here for monocytes we can give another option, bean-shaped nucleus, right? Then what about basophils? Basophils you can match with secretion of histamine, serotonin and heparin. Histamines are inflammatory substances. Serotonin is a vasoconstrictor. And what about heparin? It is an anticoagulant. Then we have monocytes matched with the bean-shaped nucleus. Lymphocytes with responsible for immune responses. And eosinophils you can match with allergic reactions. So after matching, our answer will be option C over here. Okay, so the correct answer will be option C. Right. So shall we move to our next question now? So this question is about the different blood groups. You know, in our human beings, in the case of human beings, we have an ABO system of blood grouping. And this ABO system of blood grouping, it is based on the presence of the surface antigens or antigens that are present on the surface of RBCs. There will be two antigens considered here. One is antigen A and the other is antigen B. So in the case of blood group A, you know, in the case of blood group A, the surface antigen will be A antigen. When A antigen is present on the surface of the RBC, that blood group is called the A blood group. And in their plasma, you can find antibody B. So anti-B is present in plasma. What about B group? B group will have on their RBCs the surface antigen called B antigen. So that is why they are named as B group. And the plasma will contain anti-A, antibody A. AB group will have both the antigens. Both A antigen and B antigen you can see as a surface antigen for AB group. But there will be no antibodies in their plasma. And O group on the other hand, O group will have no antigens. It is having no antigen A and antigen B. But it will have both antibodies in the plasma. It will have antibody A and antibody B. So this is how the blood group ABO system is classified. So ABO system in the case of human beings, it is classified based on the presence of the surface antigens. We take into consideration the surface antigens here for classification. We have another slide for explanation, one more slide. So this slide will give you an idea about the donors group as well as a recipient group for each blood group. So blood type is given on this side in this column. In this column we have the surface antigen. In this column you can see the antibodies present, and here A group is having B antibody in the plasma. The plasma will contain B antibody. So B antigen should not be there in the donors group. Okay, so donor group should not have B antigen, right? So A group, when it is a recipient, it cannot receive blood from B group and AB group. So only it can receive blood from A group and O group. You got it? Like that. When you consider the B group as a recipient, the second row is regarding the B group blood. Okay. So when it is a B group blood, in the case of a person with B blood group, the antibody in the plasma will be A. Antibody is A. Antibody A. So here the donor should not have antigen A. So the donor should not possess antigen A. So A group and AB group cannot be the donor for the B group blood. A group and AB group will have A antigen in it. So A group and AB group should not come as a donor for B blood group. That is why the donor group will be B group and O group for B group. B group will have donors as B group and O group. Like that, when you consider the AB group of blood, it will have two antigens in the surface of RBC. And what about antibodies? Antibodies are totally absent. So when in a recipient antibodies are absent, it can receive blood from anyone. Okay, so here it can receive blood from anyone. So that is why A group, B group, AB group or O group for AB group. So that is why AB group is considered as a universal recipient. So why AB group is a universal recipient? It is because it has no antibodies in the plasma. So when a recipient is having no antibodies, there is no possibility of a reaction here. You got it? So it is not because both antigens are present, but it is because both antibodies are absent. AB group is considered as a universal recipient. Okay. And now what about the O group here? When O group is a recipient, it can receive blood only from the O group. Because in the case of O group, it will have both the antibodies. In the plasma of O group blood, A antibody and B antibody will be there. So A antigen and B antigen should not be there in the donor. Okay. So donor should not have A antigen and B antigen. So it cannot receive blood from the A group, B group and AB group. So it cannot receive blood from A group, B group and AB group. So it can receive only from the O group. Okay. So the donors group for each type of blood you should keep in your mind. You have to by heart. Okay. So now we can answer the questions now. We have the questions on the screen now. Choose the correct option that indicate the donors group for blood group A, B, AB and O. So we can answer it very easily. So for donors group they are asking. Okay. So in the case of donors group, which is the correct option here? For A group, the donors group will be A and O. For B group, the donors group will be B and O. And for AB group, all of them, okay, then for O group only O. So the correct answer will be this first row or option A itself. You got it? So we have the answer shown here marked. So our answer will be A. Shall we move to our next question? So here we have the next question. Again, this is based on the blood group that we discussed earlier. Identify the true statements. Identify the true statements. Two antigens A and B are located on the surface of RBC, whereas the plasma contain two natural antibodies. This is statement 1. Then second statement. What you can say about the first statement? That is a correct one. Then what about the second? AB group of blood is universal recipient as it has presence of both the antigens. This is wrong because AB group of blood is a universal recipient and it is because it is having absence of antibodies in the plasma. So here, because antibodies are absent, AB group is considered as a universal recipient. So the second statement is wrong. What about the third one? During blood transfusion, there is probable clumping between the antigen of the donor and corresponding antibody present in the recipient blood. This is very much true. So this is a true statement. What about the next one? In ABO system, the blood transfusion is not recommended from A to O. So this is also true. So our answer will be option D here because only the second statement is wrong. So the correct answer will be option D. Shall we move to our next question? Yes. The next question, it is about erythroblastosis fetalis, or this is about RH incompatibility. In the case of RH incompatibility, you have to always note that the RH factor or the rhesus factor is showing some sort of incompatibility during the pregnancy. When the mother is RH negative and the fetus is RH positive, there arises an RH incompatibility, right? So here in this picture you can see the mother is RH negative and the fetus is RH positive. So here during the 9 months of gestation there is no complexity, but at the time of delivery you can see the blood vessels are breaking. So there is possibility of some of the RH antigens. You know, RH antigens are present in the fetus because it is having RH positive blood. So some of the RH antigens, they will enter into the mother's blood. So the mother is sensitized by the RH antigen entering. So RH antigen will enter from the fetus into the mother's blood. So now the mother will begin to produce RH antibodies against it. So mother will produce, mother's immune system is activated or it is sensitized and it will produce RH antibodies. So in the yellow color you can see the antibodies are developed. RH antibodies are developed. So these RH antibodies, they will attack the RBCs with RH antigen on the surface. But already the first fetus has been delivered. So here when there is a second fetus, when the same mother conceives a RH positive fetus for the second time, then what happens? Already prepared RH antibodies that are present in the mother, they can move through the placenta. These RH antibodies, they can cross the placenta, they can enter into the fetus and it will destroy the RBCs having RH antigen on the surface. So the second fetus will also have RH positive condition. So all the RBCs having RH antigen will be destroyed by the antibodies entering from mother to fetus. So here the second fetus will be adversely affected. So again and again when there is a delivery or during parturition, again there will be mother sensitized with RH antigen and she will begin to produce more antibodies. So the amount of antibodies will increase in the mother and the fetus is going to suffer. All the RH positive fetuses, they are going to be affected. And the condition can be called hemolytic disease of the newborn because here there is hemolysis or blood cells are destroyed, or you can also call it as erythroblastosis fetalis. You got it? So in which condition this complexity occurs? This complexity occurs when the mother is RH negative and the fetus is RH positive. So that is a condition. So we have the question for you. Erythroblastosis fetalis occur when It will occur when the mother is Rh negative and when she carries an Rh positive fetus. So answer is very simple. Our answer will be option A here. Is it clear? So shall we move to our next question? We have the next question on the screen. This is about the different valves. You should have a clear idea about the different valves present in our heart. Our heart is having a tricuspid valve. The right AV valve is called tricuspid, and the right AV valve is between the right auricle and the right ventricle. And between the left auricle and the left ventricle, there will be the left AV valve, and left AV valve is bicuspid, and because it will have only two cusp-like structures, and it is also called the mitral valve. So a tricuspid valve is present on the right side, and it is a right AV valve, and a bicuspid valve present on the left side, and it is also called the mitral valve. Okay, so here tricuspid valve will regulate the flow of deoxygenated blood, whereas the mitral valve will regulate the movement of oxygenated blood. And what about the semilunar valves? Semilunar valves are associated with the major blood vessels leaving our heart. You can see that the pulmonary artery is beginning from here. So associated with the pulmonary artery, you can see the pulmonary semilunar valve. Pulmonary semilunar valve, again it will regulate the flow of deoxygenated blood. What about the aortic semilunar valve? Associated with the aorta beginning from the left ventricle, there will be aortic semilunar valve, and aortic semilunar valve will regulate the flow of oxygenated blood, because always on the left side it is oxygenated and on the right side it is deoxygenated. So valves are of different types. AV valves are tricuspid and bicuspid, and semilunar valves are of two types. There is pulmonary semilunar valve and aortic semilunar valve associated with the aorta. So here we have the question for you. Select the wrong statement. Pulmonary vein carries oxygenated blood. That is true, because pulmonary artery will carry deoxygenated blood, but pulmonary vein will carry oxygenated blood. That is the true statement. Tricuspid valve will regulate the flow of oxygenated blood. Is it true? Tricuspid valve is on the right side. It will regulate only the deoxygenated blood. So this is wrong. It is not oxygenated. It is deoxygenated blood. So second statement is wrong. What about the third one? Incomplete double circulation is present in amphibians and reptiles. That is the correct statement, because amphibians and reptiles, they have a three-chambered heart. So here they have an incomplete double circulation. What about heart in fishes? Heart in fishes is called the venous heart, because always deoxygenated blood will be received by the heart in fishes. So that is why heart in fishes is also called the venous heart. So here only the second statement is wrong. I repeat, only the second statement is wrong. So correct answer will be option C. Okay, so we move on to our next question. Our next question is based on the cardiac cycle. The sequence of events beginning from one heartbeat to the beginning of the next heartbeat, that is called cardiac cycle. So in a cardiac cycle, the duration will be 0.8 seconds. And you can see the different events in cardiac cycle over here. First of all, it will be atrial systole. For the first 0.1 seconds, there will be atrial systole. Atria, they contract, and that will increase the blood flow to the corresponding ventricles by 70%. There will be an increase of 70% blood flow by atrial systole. So after atrial systole, the ventricular filling will be over. Okay. Then comes the ventricular systole. 0.3 seconds, you can consider the ventricular systole. And after ventricular systole, you can see the joint diastole. In the last 0.4 seconds, it will be the joint diastole. When all the heart chambers are in the rest condition, we can consider it as the joint diastole. So joint diastole means there will be both atrial diastole as well as the ventricular diastole. So in this picture you can see here the different systole and diastole, they have given different colors. Okay, you can see the outer rings here marked as systole and diastole. The blue color over here, it denotes the atrial systole. Is it okay? The blue color denotes the atrial systole, this portion. And what about the ventricular systole? The one marked as the red color here, that is ventricular systole. Okay, the red color here, the red band, it indicates the ventricular systole from this point to this point, right? That is ventricular systole. And what about the orange color? Orange color starting from here. Okay, you can see the orange color that denotes the atrial diastole. So atrial diastole will be from this point to this point. Okay, so all along this area you can consider as the atrial diastole. And what about the ventricular diastole? Ventricular diastole is in the ash color starting from here. Okay, so this ash color, this circle, this portion, this curve, it is atrial diastole. So I think everything will be clear for you. So we can answer the questions now. We have a question about the sequence of cardiac cycle. Remember, already before the cycle happening, the AV valves are open passively. There is a passive flow of blood from auricles to ventricles. There will be a passive flow of blood from auricles to ventricles because AV valves are open. So here 30% of blood reaching the auricles will move passively to the corresponding ventricles. And atrial systole will happen, or atria will contract only to increase the blood flow by 70%. So after atrial systole only, the ventricular filling will be over. So after atrial systole only, the ventricular filling will be over. So our answer here, atrial systole, after that only ventricular filling. Then comes the ventricular systole and ventricular diastole. So here the correct answer will be option D over here. Yes, in a cardiac cycle, duration of different events are represented here. So here you can see the duration of different events represented here. So here you have to answer some questions. Identify A, B and C. So you know A, B and C. A will be the atrial systole, B will be the ventricular systole and C will be the joint diastole. And duration of B and C put together. So here the duration of this is duration of B and this is duration of C. So B and C put together, that will form the atrial diastole. For the second you can answer atrial diastole. And what about the third question? The duration of C and A put together. So this is C and this is A. So C and A put together, that will form the ventricular diastole. That will form ventricular diastole. So the correct answer will be option A here. Okay. So correct answer is option A. So labeled as A will be atrial systole, labeled as B will be ventricular systole, C will be joint diastole, and you can also identify the atrial diastole and ventricular diastole as combined regions. So our answer will be option A here. We move to our next question. This question is about the ECG, or the graphical representation of these cardiac impulses that you receive as a result, that is called electrocardiogram. So electrocardiogram is represented over here, and you can consider the different waves like P wave, QRS complex, then there is a T wave. So you should identify what they represent in a normal ECG. In case of normal ECG, the P wave represents atrial depolarization. P wave represents atrial depolarization. What about the QRS complex? QRS complex will represent the ventricular depolarization, and at the same time you can consider it as the auricular repolarization. So ventricular depolarization and auricular repolarization is represented by a QRS complex over here. Okay. Then what about the T wave? The T wave represents the ventricular repolarization. T wave represents ventricular repolarization. Is it clear? So we have the question for you. So in this question, in the ECG of a normal person, match column A and B. So it's very simple. Atrial depolarization you can match with P wave. Ventricular depolarization and atrial repolarization, both of them you can match with the second one, or QRS complex here. Then ventricular repolarization we can match with the T wave. So the correct answer will be option A. So the correct answer is option A. Right? We move to our next question now. So this question is about the conducting system of the human heart. Human heart is having a conducting system, or there are specialized nodal fibers for conducting impulses for working of our heart. Our heart is myogenic. Our heart is working with the help of specialized nodal fibers, and these specialized nodal fibers are SA node, AV node, then AV bundle and Purkinje fibers. Okay. So here, specialized nodal fibers, they are responsible for initiation as well as conduction of impulses. So here, which is the natural pacemaker in human heart? In the case of human heart, the natural pacemaker is our SA node. SA node is the natural pacemaker located at the upper lateral wall of the right atrium, or very near the opening of superior vena cava. And what about the AV node? AV node is considered as a pace setter. AV node is a pace setter, and it is located on the lower left corner of the right atrium. On the lower left corner of the right atrium. So now we can answer the questions now. So on the screen we have the question. Choose the correct pathway of transmission of impulses in the heartbeat. So here the correct sequence will be, what is the correct sequence here? The correct sequence will be SA node, AV node, then AV bundle and Purkinje fibers. So that is the correct answer here. Okay. So the correct answer, correct option will be option B. I repeat, what is bundle of His? Bundle of His will be the bundle branches and the Purkinje fibers together considered as the bundle of His. Bundle branches and Purkinje fibers together will form the bundle of His. So the correct answer will be option B here. So we move to our next question. This question is based on again the blood vessels and the different valves. So you should have a clear cut idea of the different valves involved in blood circulation and also the type of blood flowing through them or regulated by these valves. Okay, so this picture clearly shows the location of the different valves. You can see the AV valves located here. You can see the pulmonary semilunar valve and also aortic semilunar valve over here. Okay, so we have the question. From the question we will answer very easily. Match the following columns according to the type of blood passing through the type of valve. So oxygenated blood is actually passing through the left side of our heart. So here mitral valve, then aortic semilunar valve, and then mitral valve is also called the bicuspid valve. So here PRS you can match with oxygenated blood. PRS you can match with the oxygenated blood. And what about Q and T? Q is the valve present in pulmonary artery, pulmonary semilunar valve. So it is for regulation of deoxygenated blood. And like that, the tricuspid valve, the right AV valve is also called the tricuspid valve. It is also regulating the flow of deoxygenated blood. So the correct answer will be option D over here. Okay. So we move to our next question now. So we have the next question. It is on the blood coagulation. It is on the blood clotting. You know, the process of blood clotting will initiate when there is an injury or removal of blood from vessels. So here, the preliminary steps in clotting will happen when there is rupturing of the platelets. So platelets are releasing prothrombinase, and prothrombinase will activate prothrombin to thrombin. For that, calcium ions are necessary. And once thrombin is formed, thrombin can convert fibrinogen to fibrin threads. So both for activation of the thrombin and also for the action of the thrombin, for this reaction also calcium ions are necessary. And once fibrin threads are formed, along with the blood cells and plasma, a clot will be generated, a clot will be formed. So this is how blood coagulation happens. So these are the steps in blood coagulation. Remember, for activation of the thrombin as well as for action of the thrombin, calcium ions are required. Okay, so we have the question right now. Blood coagulation, the true statements are. So here a lot of statements are given. Fibrins are formed by conversion of the dissolved fibrinogen in plasma by enzyme thrombin. That is correct. Thrombins are formed from inactive substance present in the plasma called prothrombin. That is also true. Then thrombokinase is required for the activation of the prothrombin. That is a correct statement. Then an injury or a trauma stimulates the platelets in the blood to release the platelet factor 3. That is correct. Calcium ions are required for the action of the thrombin. So all these statements are true. So our answer will be option D. Okay, we move to our next question now. So we have the next question. It is on the type of circulation present in different vertebrates. In the case of fishes, they have only 2 chambers for their heart, one auricle and one ventricle. So the circulation will be more or less like this. Okay, so always a venous blood will be received by their heart. That is why their heart is called venous heart. In the case of amphibians and reptiles, they have a 3-chambered heart. So there will be mixing of oxygenated and deoxygenated blood when it reaches the ventricle. So this is circulation applicable for amphibians and reptiles. But in the case of birds, mammals, they have a true 4-chambered heart. So they have a double circulation. So they have a complete double circulation in the case of birds and mammals. So we have the question now. This is about incomplete double circulation, because in the case of amphibians and reptiles, it is incomplete. So here, left atrium receives oxygenated blood from the gills, skin or lungs, and the right atrium gets the deoxygenated blood from the other body parts. That is a correct statement. Oxygenated and deoxygenated blood is getting mixed up when it reaches the ventricle. That is a correct statement. Then we have the third statement. Incomplete double circulation is present. It is true for amphibians and reptiles. Heart is 3-chambered. That is also true. So here all the statements are true regarding amphibians and reptiles here. So the correct answer will be option C. Okay, we move to our next question. This question is about the conducting system. Okay, so here you can see the SA node, AV node, okay, the bundle branches, and you can also see the Purkinje fibers. So what do you mean by the bundle of His? By bundle of His, we mean the bundle branches and the Purkinje fibers. Together, that will form bundle of His. So you can see the right bundle as well as the left bundle marked there. So we have the question for you. This is about the position of the SA node. SA node is situated on the upper lateral wall of the right atrium, and it is just below the opening of the superior vena cava. So here both A and B are true. So the correct answer will be option D. What about the AV node? AV node is very close to the atrioventricular septum. AV node is very close to atrioventricular septum. So we have the answer option D, because the question is about SA node. Now we move to our next question. This is about the heart sound. So regarding the heart sounds, you have 2 heart sounds in your NCERT book to learn. One is the first heart sound and other is the second heart sound. The first heart sound is heard due to the closure of the AV valve. What are AV valves? The right AV valve and the left AV valve. So by the closure of the AV valves, you can see the first heart sound. What is that? Lub. So the lub sound is heard by the closure of the AV valve, and it is heard at the time of ventricular systole. So ventricles, when they undergo a systole, when they contract, to prevent the backward movement of blood from the ventricle to the corresponding auricles, the AV valves will close. So that will result in the first heart sound. What about the second heart sound? Second heart sound is a dub sound. So after lub, there is a dub. So dubs or second heart sound, it is due to the closure of the semilunar valve. So semilunar valves will close during the ventricular diastole. So during ventricular diastole, already blood has been pumped to the main blood vessels. Through the pulmonary artery and aorta, blood already pumped by the ventricles. So it doesn't want the blood to return or come back to ventricles again. So here, for that, there will be closure of the semilunar valves. So semilunar valves will close to prevent again the backward movement of blood from the corresponding blood vessels into the ventricles. Right. So that will cause the second heart sound or dub sound. So doctors are able to hear lub dub, like that. The sounds, heart sounds, major heart sounds are lub and dub. So here we have the question for you. The second heart sound or the dub sound is associated with the closure of the semilunar valve. So the correct answer will be option B here, right? So it is not because of the tricuspid or bicuspid or not because of the AV valve. The correct answer will be option B over here, right? So this was our last question. So with this we conclude today's session of NEET crash course. Tomorrow I will be back with another chapter from second year. Until then, it's goodbye from Stephen Classes.